So that's what we're going to be talking about today. If you do have questions more about my research program, please let me know. But today we're going to be taking a 3,000-foot view. We're going to look at the past 5 years and the progress we've made as an industry, but also as researchers. And then what's coming, what's the next 5 years. And so I hope today we leave with a shared roadmap. So my talk, as you can determine, is separated into 2 parts: what we've learned the past 5 years and the roadmap for the future of the next 5 years. And that roadmap was really something that ARS wanted to put together so we could continue this coordinated effort that was aligning industry concerns with the science. Here we have at least what we've learned is identifying key marker compounds, progress in risk assessment, prevention and mitigation in those early days, and then also where we've come from aligning the science with industry concerns. And then looking forward, building that baseline database, I'll touch on this later, establishing great threshold levels, rapid affordable detection tools, and atmospheric modeling and prevention. So let's start at the beginning. While researchers in Australia had made incredible progress since 2003, when this first issue came to light, when one of the first instances of smoke taint came to notice. But since then, it really didn't come to the US, or at least into researchers' minds and industry minds, until 2016, maybe 2008. I know there was a lot of folks worried about some of the fires of 2008. But in terms of research, it really didn't hit until about 2016. And at a federally recognized level, not until 2020. We couldn't ignore it anymore after the 2020 wildfires. And I'm sure I do not have to remind anybody in this room of what was 2020. So this was one of the last years of my PhD. And although I was primarily working on grapevine red blotch fires, my PhD switched to smoke taint and smoke impact. So in 2020, over 6 million acres burned in the Western United States and just over 4 million in California alone. This economic loss was estimated like $3.7 billion for the industry in direct and indirect impacts. And I really want to say impacts here and really make sure we define what smoke exposure is versus smoke taint. And those are really key concepts to differentiate. Smoke exposure is just that, when smoke is lingering in a vineyard and exposing the grapevines. Smoke impact can be any range of impact on grape and wine composition, and smoke taint is the sensory changes and perception in the wine. So we need to make sure that we define those clearly before we go into the rest of the research. And as you've already heard, one slide ahead. So let's go a little bit through what smoke exposure is, just briefly. So when smoke lingers in a vineyard, it is rapidly absorbed by the grapes within minutes to hours. So really, if smoke is present, it is absorbed by grapevines, both the leaves and the grapes themselves. This is then transported and moved around into the grape tissues, mainly by it being formed into a glycoconjugate. And so we now have a free volatile phenol, adding a sugar group onto it and changing it both physically and chemically. So now it's a non-volatile compound, it's water soluble, and that's how the plant is able to move it around into different cell tissues and cell components. The real issue comes with fermentation. Now during fermentation, we have enzymatic activities that can then cleave those bonds and doing hydrolysis reaction that can reform those volatile phenol odoractive compounds, as you can see here. So we now know that it's really the presence and the balance of both of these, as well as the newly identified thiophenols, that give the perception of smoke taint within wine. We also know that our saliva has enzymatic activity that can also cleave these compounds in mouth and giving or related to the ashy aftertaste. So you've already heard it's a complicated issue and what we're working with. We know that it can render our vintage unmarketable or unsoluble. We know generally what we're looking for and the key issues of volatile phenols, thiophenols, and glycoconjugates. And we also know that the frequency and intensity of wildfires is only increasing. So wildfires are not a future risk. They're here for present reality right now, and our science has to catch up fast, and it will continue to catch up fast. And the progress that we made in the past 5 years is we now know what we're looking for. In 2020, many growers and winemakers were still analyzing for methyl glycol and glycol only. We now know that that is not the complete picture. We know that we need to be analyzing both volatile phenols and those glycoconjugates, because as I said, volatile phenols as a detoxification response in the plant are sequestered and formed into those glycoconjugates. And in some of my studies, I've seen where we had within 5 days, increased volatile phenol pool decreasing to almost baseline levels within those 5 days. So it's really important now that we measure these glycoconjugates. And with Elizabeth Tomasino's work and Tom Collins, we also know that thiophenols play a large role in the perception of smoke taint and as a composition marker in grapes and wines. These methods are still maturing in detection and quantification methods, but we now know that this list that I show here is dynamic. It's not a static list, and researchers are continually trying to make sure we know what is causing smoke taint and what is attributed or correlated with smoke exposure. So here we have 7 volatile phenols. We have 4-methylglycol, glycol, and creosols, as well as syringol. What I highlighted here in red is what research now has pointed to is correlated and associated with smoke taint. Rather, where we have syringol, 4-methylsyringol, they're not really well associated, not at all, with perception of that finished line of smoke taint. And the same thing on the right, we have 6 glycoconjugates. Syringol gentiobiocide is a really important smoke marker compound, but work out of Australia as well as in the US has shown it's not attributed to smoke taint perception. So while we can say, yes, we have smoke exposure in our vineyard at a high level, we might not know how that correlates with smoke taint. But with measuring creosol rutinoside and glycol rutinoside, we can have a bigger picture. And like I said, this is dynamic, especially as we have thiophenol detection methods maturing. Now, how do we measure these compounds? And this is really the gold standard right now, and I'm sure this is not new to anybody in this room, but LC and GC chromatography methods are the primary way we measure these compounds in third-party labs, but as well as in research and universities. They're highly sensitive and precise. We can get down to parts per trillion, if not parts per billion, if not parts per trillion levels, especially when we're thinking about thiophenols. However, they're expensive, they're time-consuming, and they can have long turnaround times, as I'm sure people are aware of during 2020, when some of those turnaround times were 3 to 7 weeks. It's very impractical when you're trying to make harvest decisions, and we know that. And ETS, for instance, has worked incredibly hard to increase or decrease their turnaround time so you get reliable information that you can make a competent decision. But the reality is it's still days. And so researchers started to look into how they can increase the turnaround time, but also have more correlation to the wine outcome. And this is where nanofermentations came into play. And this is really coming from research with Anita Oberholzer, Tom Collins, and Elizabeth Tomasino on how we can simulate that winemaking at a small scale, have relatively faster and lower cost alternatives, meaning you still have days where you have to do this fermentation, but now you can actually have the finished product. You can look at the wine, not just the grapes, both chemically and sensorially. So we're still looking, and this is really what drove, I'm going to talk about the roadmap a little bit later on, is driving the need for something that can give you values or results within that same day through testing. And we'll get into that a little bit later. And as we've already mentioned, it's complicated. There's so many factors that affect smoke exposure and risk assessment. Here's just a few I've put out here. Fuel type, for instance, what's burning in the fire itself. Repeat exposure, wind direction, the distances traveled, which you'll hear a little bit later here from Mike David. And also the cultivar and ripeness rates. You change one of these and you have a different picture. So that's what we're up against. And as a team of researchers, we are now trying to understand how we can model this and give you the best prediction of smoke impact in your vineyard. And some of the progress that's been made in the past 5 years is really focusing on what does normal look like? What does your normal level in grapes look like so you know what elevated looks like? And that came from the baseline database or baseline data. And so there's several research labs as well as wineries and internal labs that have gathered what natural levels of volatile phenols and glycoconjugates are across cultivar, season, vintage. And what we know now is cultivar is the dominant driver, meaning the differences between Syrah and Cabernet are going to be the largest differences you see, not vintage to vintage. And that's really promising now because we can focus on one target or one variable. Next is sensory thresholds. I'm just going to touch on this a bit as you're hearing from Elizabeth Tomasino that can go much more in detail about this. But there is a difference between wine style and the consumer. We really need to understand that as we discuss this issue with our consumers, that they are different per person. And again, Elizabeth touched on this, but we now know around 20 to 30% of the population does not perceive ashy aftertaste. So that is important. We're actually providing these wines to our consumers. And then atmospheric models, and I'll get into a little bit of my research on atmospheric modeling, but this has really picked up starting in 2020, knowing that we need to know what's happening in the atmosphere, how that smoke is moving, what's in that smoke, and how it might degrade over time as it goes from point A to 50 miles further on. So we're not there yet with vineyard specific or real decisions, but we're getting there, and the progress has been immense. So I want to just touch on a little bit of research that went on in my group in 2023. And this came from a collaboration with Tony Wexler at UC Davis, as well as the Desert Research Institute at in Nevada, Reno, my hometown. So what we used here was passive sorbent samplers, and they're as big as my pinky. They are pretty much a sorbent body that can capture compounds in the atmosphere, even in your breath. So anything that's volatile, it can capture and absorb onto its body. The best thing that we're looking for here is that they're very cost effective. About one of those sensors is $150, but it actually increases in affordability when you have more sensors. They are easy sample prep when you compare grape sample prep, all the phenolics you have to get rid of, and the liquid extractions and the sample prep versus here, you just have a sorbent tube. You thermally desorb it or remove it thermally with all those compounds and then run it through GCMS. However, as I just mentioned, still have a GCMS. Still have to have that expensive equipment and analytical costs. I will say that things are looking a little bit more promising in this field as Tony Wexler has now purchased 8 portable GCMS units. Didn't really think that those existed except in the military, but yeah, now we have 8 portable GCMS units that we can actually measure this in the field. So that's ongoing work here. But what we wanted to do in 2023 is correlate the level of volatile phenols, the specificity of looking directly at volatile phenols, not total VOCs, and correlate that with composition in grapes and in wines. So that's what we did. And here you just see 4 points. We smoke exposed grapes at 4 different levels, a control level, a moderate, arbitrary low, medium, and high. We modulated this based on the mass of the wood we were burning. And here you can see on the left, we have the mass of the volatile phenols. In this case, it's glycol in the sampler, the concentration in grapes, and the concentration in wines. And then we also did a regressive analysis where we have the concentration in grapes correlated with passive samplers. And you can see this is almost a perfect fit, 0.9999. Now, I want to give a little bit of a caution. This is 4 points. This is not a large linear curve. There's only 4 points. So this is very preliminary data. So this fit is a very accurate or a very strong fit, but how accurate is it? So we wanted to back calculate this and see how it was able to predict based on the passive sampler values, grape concentrations. And here we had below 5% error for grapes and below 10% error for wines. So like I said, preliminary results, but promising results on how we can push this forward and use this technology in a vineyard setting. And so now this has actually been grown into a larger project with Tony Wexler and how we can put these passive samplers in a larger set in a vineyard and then correlate this with not only impact in wine and sensory impacts in wine, but grape composition. So that's just a brief view of what we're doing in terms of risk assessment and field assessments. In early days of prevention and mitigation strategies, a lot of researchers in this room have looked into barrier spray technologies. If you can coat your berries with a spray and either reduce the amount of volatile phenols coming in or eliminate it altogether. And this is really coming from blocking technologies. If this barrier spray is actually able to block the volatile phenols being absorbed into the grape, rather just being another absorbent body that you might have to remove later on. There's also genetic approaches, whether that's breeding, but researchers in ARS are also looking into some CRISPR editing, so how we can understand a little bit of the mechanism of smoke taint in the berry and those biochemical responses, and how we can maybe create either resistant or less susceptible grapevine. In the winery, we have enzyme-based treatments, and there's 2 labs I could think of that have been really working in this. And one of them is in ARS, and it was really interesting, a lot of the work he was doing. He was looking on microbes that are already naturally present on grapes, understanding a little bit more about their biology, and seeing that they can actually consume guaiacol and syringol naturally. So now how can we use this technology and make another yeast strain? So now you have a yeast that can naturally produce or consume these volatile phenols during fermentation. And they form non-toxic, already present compounds in your wine. So you're not adding anything new to your wine matrix. Also looking into absorbent technologies, zeolites and MIPS. And this comes actually from a collaborator of mine at ARS who's looking into MIPS specifically and reverse osmosis and understanding what current technologies are already out there that can reduce this impact of wine. But right now, there's no tool that is bulletproof. We have strategies that can work together in a synergistic manner, and they might complement each other. And so what came out of this workshop too is, what do we know now? How can we build a decision tree? And where do we need to go to the future for mitigation and prevention? So I'll touch on this a little bit for the work that I've been doing with Dario Cantu at UC Davis, as well as Roger Tillmany, who I mentioned was doing some of the CRISPR gene editing. And so here we took Pixie grapes. They are one of the most fun cultivars I've ever had to work with. They are a dwarf plant of Pinot Meunier. And so they're for research purposes only. And I say that because they can grow clusters all year round. We can actually do our experiments all through the season rather than that 3 month chunk. And if you look Pixie plants up in Google and Wikipedia, you can see a shoot with all different stages of maturity. It is absolutely fascinating. And that's for us, we can actually do really interesting work on these plants and see how it can translate then to Cabernet Sauvignon or Merlot. So here, he already had work looking into doing the full genome sequence of Pixie plants and then also correlating that with Pinot and Merlot. But here what we did is we had 2 gas exchange desiccators and we impregnated one with smoke that was produced from oak and one that was a control chamber. And we also had monitors, the purple, actually no, those are air thingies inside monitoring carbon monoxide, carbon dioxide, total VOCs, temperature, humidity. We then sampled berries 24 hours after exposure and then 72 hours exposure for RNA sequencing and well BP analysis. And what we found is that there's 12 upregulated glycosyltransferase genes across the samples that peaked at 24 hours and slowly decreased at the 72 hour. And this is interesting because this tells us that it's a rapid transcriptional response that is happening in the grape berries to glycosylate these volatile phenols. We also saw this correlated with the decrease of volatile phenols and the increase of glycoconjugates. And so we now know that these 12 upregulated glycosyltransferase genes are a potential target for future studies, whether that is mitigation or prevention. And one way we're looking at this is, can we reduce glycosylation overall? Can we reduce glycosylation? So now researchers, we're only working with volatile phenols where we're trying to build mitigation strategies. So like I said, they're both physically and chemically different, making it very hard to target our strategies. The other way we're thinking about this is forming triglycosides. In some of the research and tomato research, we found that it is irreversibly bound and potentially can reduce the perception of smoky or ashy flavors in a wine. So we're looking at both avenues, but as you probably know, anything genetic modifying or breeding takes long-term strategies, long-term. So I just talked for about 18 minutes, very roughly about 5 years of work. I did not touch on everything. So please, you're in for a treat for hearing from the other researchers, but if you have any questions about my research, please let me know. I only shared about 2 of the topics here. But let's look forward. What are the next 5 years look like? What lies ahead and how can we continually work with industry to align our goals? There's more scientists now dedicated to this issue than 2016, even 2020. We have atmospheric chemists, microbiologists, engineers, all working on this that are not in the wine sector or have not worked in the wine sector before. So this is remarkable. We're pulling in really great minds to come together and make immense progress. So let's talk about how this roadmap was developed. So this was a 2-day workshop that was held in Corvallis, Oregon, with 50 researchers, industry members across Oregon, Washington, and California, both ARS or federal government scientists, as well as researchers at universities. We had a small format. We really wanted to keep it to 50s just so that we could really increase the amount of discussion that was occurring. We had a format as well where we had breakout sessions. We grouped it into topical sections for a reason, so that we could have 5 topical sections and 5 groups. We also had then people rotate with those groups for cross-pollination. So you didn't just share on one topic, you were able to share your learnings and your knowledge on every, or at least 3 avenues of this field. Those 5 topical groups were, how do I know what's normal? Baseline database. What should I be worried about? And really understanding threshold levels, both grape threshold levels that lead to impact in a line, and then sensory threshold levels and understanding consumer differences. How do I test quickly and cheaply? And this was looking into rapid detection tools. Can I see smoke coming? And this is atmospheric modeling, which you'll hear later on. And what do I do about it? Prevention and mitigation. So we're going to walk through these one by one. Let's first look into building a baseline database. So the baseline data exists. Now it's bringing it all together. How can we bring this knowledge together to form a useful tool, user-friendly tool for the industry to know what normal looks like? And so, as I said, we know these samples exist. The core problem is understanding if we can bring together samples that are measured slightly differently. There's slightly different analytical methods that are done in each lab, and making sure that we can pull that data together in an accurate and robust manner. And so this actually has been coming through Australia in a ring test format, where we have samples sent to various labs that are all identical samples and also reproducible, but they're blind to the researchers and the labs. So then we can actually compare inter-lab comparability and also the reproducibility in the lab. And so we're going to try to continue this. And the solution, I'm just going to give you a glimpse. This is something that's underway, but we're not anywhere near actually creating a web base at this point. Right now, we're just trying to work with partners. So I really want to highlight the values you're about to see are just for visual purpose only. So you can understand how we're trying to align a user-friendly format that you can use to understand what elevated looks like. So we really want to have a database where you can see at the bottom right-hand corner, you can modulate based on cultivar, season, and location. And then you can see what Syrah and Vineyard Block in Oregon looks like versus Syrah in California looks like, if those values are actually similar. And we want to make sure, as you can see in the top, 848 samples, we need to have thousands of data points to make this a very robust and rigorous database. So that comes with help from industry and knowing that we can come together and build this together. So the hope is, at least with ARS, is to have this by next season. So now let's change gears and look into establishing grape pulp threshold levels. So this is a very complicated strategy and has been attempted in other formats, such as terpenes, if you can understand like grape-derived terpenes and how they progress through winemaking. It is a very complicated picture, as you're seeing, working with very biochemical processes that could change based on yeast selection, how long you macerate. So we really want to understand this further and at least give you threshold levels. Really defining this, and this is just the 3 names, of defining these threshold concentrations by cultivar. These will not be universal and they'll likely change with wine styles, but really just giving you a gauge to work with, then to make your decision on what wine style you might want to target now, knowing that X, Y, and Z causes increase. Investigating phenological timing and compound accumulation. This is really key because we know that phenological time, or if a grape is in pre-vibration to harvest, the absorption changes and the susceptibility changes, but we don't know that fully yet. And based on cultivar, it was always thought early on that pre-vibration was less susceptible. We now know that that's not true. We had case studies in Australia, as well as in one of the studies I performed in Santa Barbara, where we had peppercorn size or pea size berries that still had impact. So we know that's not true, but now we need to know the full picture, cultivar, and through the full ripening stage. And finally is building that decision matrix deliverable. So with each one of these targets, our goal is to bring something back to the industry where you can sparse through the data and all of the research and make actionable decisions with confidence. And now getting into more rapid detection tools. This is the area with the most immediate industry payoff I can see. it's a lot of where I'm focusing my work in the next couple of years is rapid detection tools and how we can change those values to be within days, if not the same day. And so industry was really pushing to have a test that was less than $50, where you can have results within 30 to 60 minutes. And as researchers, we understand that that is something that is very valuable, but it also comes at a cost with specificity and selectivity. If you want a rapid test, it's going to be less sensitive and less selective, but it will be rapid. And so now we go into tier 2, where you might have a little bit more specificity. This is something you can see near in the benchtop. For instance, some of the spectroscopic devices, such as UV vis or Raman fluorescence, can be about $10,000 versus a $300,000 GCMS. So this could be a little bit more affordable for some of the growers and wineries in the area, but it would still be more of a tier 2. This would be able to bridge the field and lab values a little bit more, but the gold standard will always remain as LC GCMS of actually bringing these samples into third-party labs or researchers to analyze accurately and precisely levels of volatile phenols, biophenols, in lactic conjugates. The reason and the purpose behind this is to reduce the amount of false negatives that go into the laboratories or LC and GCMS labs, and that overburden or increase the amount of turnaround time in those labs. So if we can reduce the amount of false negatives where we know, okay, this is not impacted, we know it's safe, we can still harvest and make our decisions rather than sending that to the lab. Really, this comes into then how we can standardize analytical methods as well. And I mentioned this briefly already of a national ring test. We are not going to have every single lab doing the exact same analytical procedure, but how can we make sure that they're all robust and rigorous and that are producing similar results? And we've already made great headway on that. So there's already been a lot of progress on that front, but just the continuation of that effort is needed. Developing rapid screening methods is really tier 1 and tier 2, giving something that is a product that can give you results within an hour or the same day. And then finally, and this is most importantly, how do those results compare to wine sensory outcomes? It's great to know levels and composition, but how do we know if that's really impact or taint? So that's the kind of final picture there, is always coming back to the end product and what are the consumers can have. And priority 4, we're looking to more atmospheric modeling and real-time exposure. So we're going to hear a lot about this in a little bit from Michael Klingman. But let's just go over what the workshop went into and understood what we needed to target. And this came into what already exists is we know that we have NOAA websites and these models that can look into plumes and the transport of plumes over time. But we really needed to get vine level information. We needed to know what was happening at a block level. So not really looking at what VOCs or total VOCs are, but specific composition of volatile phenols in the smoke. And this is really just that integration between field sensors and then also. So what came of this workshop is atmospheric chemists and other chemists realizing that we needed vineyard level exposure characterization, and not just PM 2.5 and CO and CO2, but also volatile phenols at that vine level. How smoke was transporting through the air and also chemical composition, but chemical photochemical degradation. How are these compounds changing not only through time, but through distance? And finally, chemical surrogates for smoke exposure. Could we use other markers, like, for instance, potentially ozone, carbon monoxide, to relate with more the harmful smoke exposure compounds? Can we use more easily measurable compounds like ozone to correlate with impact in the grapes and wines? And finally, the last priority of this roadmap is prevention and mitigation. And by definitely not one of the most important ones we have to focus on as well. But in order to make progress in this area, we need to know a lot more about the other 4 priorities, especially mechanistic, biochemically what's happening in the grapes when smoke exposure happens. And so we see this in 2 different ways, what is happening in the vineyard and what is happening in the winery. In the vineyard, it's really understanding more, and this is what I'm getting into the mechanism behind, is phenolic binding research. It's understanding how these compounds are not only entering the grape or diffusing through the cuticle wax and into the grape area itself, but then also how they're actually glycosylating these compounds and other potential pathways that are happening. Really looking into collaborating with food industry. And this is coming in, we know now that there's plenty of barrier sprays or food or edible sprays that can be applied to grapes that can then be used in the winemaking process. So there's research going on in that way as well. And like I said, more barrier spray technology, looking into novel biopolymers or food grade barrier materials that are more into blocking rather just than absorbing the compounds. And then, like I said, genetic resistance, how can we move this forward? This is a long-term strategy, 5-10 years down the road. So as well as it's important, we need to also focus on other prevention strategies. In the winery, we're thinking of a multi-tier winery strategy. Since we are working with various chemicals that are different physical and chemically, we need to think of a multi-tier strategy. For instance, if we're able to cleave all these volatile phenols, glycoconjugates, into their free form, now how do we remove them? So maybe enzyme-based reactions partnered with reverse osmosis or some binding technologies. Selective absorbance is where we come to this, of how we can actually remove these, but selectively. We don't want to be removing our anthocyanins or terpenes or favorable organoleptic compounds. And then biological degradation. And I already kind of mentioned this with one of my ARS colleagues, Devin Coleman-Durr, who's looking into how they can use naturally great microbiomes in modulating yeast enzymatic activities to consume guaiacol rather than just convert it. So with this and with closing, wildfires aren't going away. But our ability to understand, predict, and respond to smoke exposure grows every season. And this roadmap is our best chance at making the growth coordinated, targeted, and fast enough to matter. And so I'll leave you with this of understanding baseline database, grape threshold levels, rapid detection tools, atmospheric modeling, and prevention and mitigation. With that, thank everybody that has worked with me in the past 4 years, and thank you for all of your attention.
Any questions? Plenty of time. We're here to discuss today.
Is there any cross collaboration with other industries? They might be farther ahead and ops and other.
I would say in the grape and wine industry is the furthest along in terms of industries, but there's cross collaboration within the same industry with other countries. It's yes.
You mentioned like the glycoconjugates are very water soluble. So maybe this is a naive question, but with that smoke also settling over the soil, is there any impact with perhaps like repeated exposure over years, or does it not matter if literally the exposure is just on the soil and leaching down? Or is that?
So far, what we've seen is that there's no impact from repeat exposures on the longevity of your vineyard. It is mainly just year to year. And mainly the impact we see at a vine level is just photosynthetic abilities of vines. But so far, we haven't seen that longevity impact.
The different compounds get glycoconjugates at the same rate, or prefer one shape or size to another?
That is still ongoing work and that we're trying to understand. What I can say is that the disaccharides or adding 2 sugars is the most preferential, and that's your Gen 2 biocides and routinicides that form versus just guaiacol glucose. So we know that the monosaccharides are less abundant than the disaccharides. But in terms of what syringel does versus guaiacol, that's still a little bit of limited research that we have right now.
And is the fermentation step, are these, I guess the enzymes that are breaking down these glycoconjugates, are they product inhibited? Like, or so the mid fermentation, you're not blowing off very much and that they get lysed later on, or do they?
It goes on through the whole fermentation process as long as you have those. We do know now that aging is not a portion of that. We thought that previously that if you had a wine that from 1 to 5 years, all of a sudden smoke taint came back. And that's not because they are hydrolyzed any further. It's just because your wine is changing in a matrix. You're having a young fruity wine to something that's been aging. So the perception is different.
But with some of the like aromatic compounds, those are product inhibited, right? With a high concentration of sugar, then the enzyme isn't as
Oh, I see
effective.
what you're saying. Yeah, no, not in this case. We have pretty much any successful fermentation is really going to have these released and the
So you could
fermentation
use your
process.
little tube as like a fermentation bung, basically, and the fermenting gases coming off of the fermenter.
You could, but it's gonna still have that pool of glycoconjugates, and we know now that those are more important than sensory than just the volatile phenols. The volatile phenols are a picture, but it's also everything that's also water soluble that's released in your mouth.
Gotcha.
What cultivars are you finding the most sensitive or susceptible? And are there some cultivars that are really resistant?
So still ongoing work, but I think for right now, what we can say is I'm blanking on the cultivar, Petit Verdot. Yeah, Petit Verdot. I was gonna say that one's something that we're kind of seeing as like a sentinel vine potentially. And I've seen growers more have this knowledge. If they have a Petit Verdot in their vineyard, they might say, test that first to see if there's anyone else have impact. But I know Pinot Noirs and Sauvignon Blanc from the big ones. Really? Okay. Yeah. Yes.
So yeah, you mentioned like during, when you're using the pixie grapes as well, that you're using smoke generated from those. Is there any potential studies as well in looking like in correlating with baseline studies across different regions where different material is learning? Is there any research into looking at differences in upregulation amongst the different types of smoke produced in those regions, as well as different glycosides produced?
Absolutely. And that's actually something that we're starting more now because we did a partnership with Chile. And in their industry, they mainly have, because it's a pulp and paper industry, they have a lot of pine and eucalyptus. So it's very different. So there's two avenues I can see this being important. How does that sensorially change what smoke taint looks like? Because it is going to change. But how does that also relate into our impact and what we can assess? What does syringeal gentubicide elevated look like when we have pine burning versus oak grass? So it is an ongoing avenue of research we're looking into. But what I can say is when a wildfire happens, every wildfire has its own signature. And it's really hard to determine what is the major components burning and what's that correlating to impact. And you'll hear from Mike, every wildfire has its own little signature. Where I see this maybe being really important is for scrub fires, where they do have maybe a single species growing it, or at least maybe not species, but type of plant, where it's grasses and brushes versus oak. And that's kind of where we're targeting now. Yeah. Yeah.
Which genes are being experimented with CRISPR analysis? And do you have a timeline on when any of those products might be available for growers?
Yeah, so right now it's glycosyltransferases, but I would say as we're doing more and more of these studies, that target is dynamic. For us, it's glycosyltransferases because we know they play a role. But what I've learned from a lot of geneticists I work with is if you knock out one, you can also knock out a lot of your terpene glycosylation, which is grapes. So that's why we're trying to maybe target the tripeptide formation. In terms of availability for growers, years. And I think it's also then the adoptability of GMOs by the industry. So that's going to be kind of that hurdle we would have to come across at that point. So breeding might be where we need to target in the future. Yeah.
On your atmospheric research, are you doing any predictive modeling for growers that could use time and distance from a fire source to even worry whether there would be exposure, or even if there's smoke in the air, but you don't have to worry about it because of time and distance from the fire source? Or
I'm going to let Mike answer that in about 4 hours.
Oh.
He has a great presentation on just that.
Right.
Yeah.
Good intuition.
Yes, it is great.